Okay, so the first part of this project is going to be the mechanical digestion of this piece of bacon. So there's a couple parts of mechanical digestion. First, of course, is the chewing in the mouth, which the teeth, the tongue, you chew the food into like mushed, basically balls, and then you swallow them, and where it goes down your esophagus. And then in the stomach, something called propulsion happens where the muscles in the stomach like contract and break it down even farther. And a similar thing happens in the small intestine with segmentation. Okay, so this is going to be the chemical digestion of this piece of bacon in the digestive system. So first will be the digestion of the protein in the bacon. So the digestion of protein starts in the stomach, where enzyme pepsin breaks it down into polypeptides. This is a polypeptide. It is just a chain of amino acids. So, it passes into these polypeptides pass into the small intestine. This is where the enzyme trypsin comes in. This is trypsin. It comes in and it cuts it into smaller polypeptides. So right here, we have our smaller polypeptides, right? So then enzyme peptidases, which is produced by the pancreas. Trypsin is also produced by the pancreas. So peptidases comes from the pancreas into the small intestine, and it breaks down these polypeptides even farther until they're just single amino acids. So then you just have single amino acids in the small intestine. This is where it can absorb straight into the bloodstream through the small intestine, where it gets transported to cells and used. Okay, so this is the digestion of the carbohydrates in this piece of bacon. So the digestion of carbohydrates actually starts in the mouth, and where the enzyme amylase, which gets secreted by the salivary gland, and it's in saliva, breaks down polysaccharides, which is what carbohydrates is made of. This is a polysaccharide. It's just multiple, and it gets breaking down into disaccharides and trisaccharides by amylase. So for this, I'm going to break it down into two disaccharides, which are called maltose. So then these maltoses travel through the esophagus, down the esophagus into the stomach. Here, chyme, which is a stomach acid, mixes with pancreatic juices, and the pancreatic juices also contain amylase, which continues to break down polysaccharides into maltose. And then it makes its way into the small intestine. Here, these disaccharides are broken down into monosaccharides, which are just singles, by maltase, sucrase, and lactase, which are all secreted by the small intestine. These monosaccharides are then absorbed into the bloodstream through the large intestine, where it gets transported to cells all over the body for use. And this monosaccharide is glucose.
So now I'm going to talk about the HCI, which is the hydrochloric acid in the gastric juice, breaks down the food, and the digestive enzymes split up the proteins. The acidic gastric juice also kills bacteria, and the mucus covers the stomach wall with a protective coating, so it kind of just helps you from when you eat not like having holes in your stomach. So we're going to talk about how lipids or fats are digested and absorbed into the body
from the piece of bacon.
So they're usually like triglycerides, which consist of a glycerol unit thread and 3 fatty acids, and they are non-polar or water-fearing, so like they don't combine with water, and they're hydrophobic and don't mix with water. And when we eat fat molecules, an enzyme called lingual lipase is released from the saliva of the mouth. And it's not activated in the mouth. And then the fat molecules in saliva are swallowed and enter the stomach, right there. And the highly acidic stomach environment activates the lingual lipase enzymes that are joined by the gastric lipase enzymes that are released from the stomach. So these. And the enzymes begin digestion or breaking down of fat molecules by breaking some of them into disaccharides and a fatty acid. And that happens to about 30% of the triglycerides. So they digest them, and then they go into disaccharides. And then some of them remain the same. And then now the fat molecules enter the small intestine right there from the stomach. And because the fats are hydrophobic, they enter as large fat or lipid molecules, and they need to be broken into smaller droplets through emulsification, which is the breakdown of the large ones into the smaller droplets. And they're broken down by bile salts and phospholipids,
which are produced in the liver and released from the gallbladder into the small intestine.
Which are hydrophobic, which are water-fearing, and hydrophilic, which are water-loving. And so the hydrophobic region of the bile salts position themselves around the hydrophobic fat, forming the emulsion droplets. And now the pancreatic lipase enzyme released from the pancreas, right under the stomach, into the small intestine has better access to fat and can digest and break it down to mainly monoglycerides. So that pancreatic lipase go in there and change it into those monoglycerides and fatty acids. And then they associate with bile salts and phospholipids to form the micelles. So you put those around. And these are much smaller than the emulsion droplets. And then the micelles transport the monoglycerides and fatty acids to the membranes of the enterocyte cells of the small intestine, where they are absorbed across the cell membrane into the cell. And what's inside the enterocyte cell, the fatty acids are reattached to the monoglycerides to form again those triglycerides. And the triglycerides, along with other lipids, are then wrapped with proteins then be made as lipoproteins called chylomicron on the other side of the enterocyte cells. These chylomicrons are too large to enter into the blood capillary walls, so they enter into lacteals, which are lymph capillaries, and the chylomicrons are then through the lymphatic vessels and eventually enter the bloodstream, transported to the liver, the fat tissue, muscle tissue, and other tissues where they are stored and used by cells.